Hello everybody, let's start with a short introduction into the muscle tissue. The muscle tissue is contractile and is excitable. That means it can generate and propagate action potentials. The intermediate filament of muscle is desmin, and the embryonic origin is mesoderm or mesenchyme. The muscle tissue occurs in two forms, as smooth muscle or non-sarcomeric, where the contractile apparatus is not arranged into repeating units called sarcomeres, and striated, that means sarcomeric, where the contractile apparatus is arranged in repeating units called sarcomeres. And this occurs in two forms, again, either skeletal, striated muscle, or cardiac, striated muscle. Let's discuss the skeletal striated muscle first. So if we proceed from macroscopic to microscopic view, you can see that the outer aspect of the whole muscle is covered by a layer of connective tissue called epimysium. Inside we will find bundles of muscle fibers, each of these being surrounded by a connective tissue layer called perimysium. Bundles of muscle fibers. And each of these bundles contains individual muscle fibers. Each muscle fiber is surrounded with basal lamina. And there is connective tissue among the muscle fibers called endomysium. So this is endomysium, connective tissue. And these are skeletal muscle fibers. Surrounded by basal lamina. Surrounding each fiber. Now if we have a more closer look on these individual muscle fibers, in the longitudinal section you will see that these fibers are multinucleated. They have many nuclei. They are always on the periphery. There is this basal lamina layer. And there is a pattern visible in optical microscope consisting of dark and light striation. etc. So we have the cell membrane here. You have the basal lamina. Together they are called sarcolemma. We have multiple nuclei. They are always on the periphery. And we got this pattern of dark bands, which are called A bands. A like anisotropic, and lighter I bands. I means isotropic in this context. In a cross section, the skeletal muscle fibers appear like that, with the nucleus on the periphery, just beneath the sarcolemma, and we can see the myofibrils, which are associations of the contractile filaments. The inner cytoplasm is called sarcoplasm. The skeletal muscle fiber is a multinucleated structure called syncytium, and this term reflects its embryonic origin, because during ontogenesis the origin starts with fusion of multiple cells called embryonic myoblasts, that undergo fusion into a multinucleated structure which still has the nuclei in the center. It's called myotube. And during differentiation, maturation, it becomes the skeletal muscle fiber as we know it. And the nuclei migrate to the periphery, and the cross striation pattern appears. There are some residual embryonic cells that are here persisting below the basal lamina, and these are called myosatellite cells. It's believed that they have potential to contribute to healing of muscle tissue to some limited extent. This is the basal lamina. Now let's have a closer look into this pattern of dark and light bands. There are glycoprotein plates called Z-lines, into which actin microfilaments are inserted. Well, on both sides of these Z lines, of course. There are also thicker myosin filaments with their heads interacting with actin, binding to actin. So this is myosin. The myosin molecules are stabilized in the center with proteins called amyline and the sarcomere ranges from one Z line to another Z line. So this is a single unit of the contractile apparatus called sarcomere, and this repeats again and again many times. A typical dimension of a relaxed sarcomere is 2.5 micrometers. Now there's a band where we have only myosin. This is called A band. Then we got a region consisting of overlapping actin and myosin. This is the A band that we already know, and we got a region with the Z line in the center that consists of actin only on both sides of the Z line. This is the I band. The structure of the actin is more complicated. It consists of many units of a globular protein called G, or globular actin molecules, and that are arranged into a double helix. So these are individual G-actin molecules polymerized into a double helix, which is called fibrillar, or F actin. So that's one of these microfilaments symbolized here. Actin, the F actin, has got binding sites for the myosin head, which is an enzymatic activity of an ATPase. However, the binding of the myosin to actin is blocked. It's blocked by another protein that is wrapped around the actin like a ribbon, and this blocking protein is called tropomyosin. So the myosin cannot bind directly to actin as long as this interaction is blocked by tropomyosin. However, there is another protein called troponin with 3 subunits. 1, 2, 3, So this is troponin with 3 subunits called T, I, and C, troponin. And briefly, when calcium cations will bind to the C subunit of the troponin, this will cause a change in the spatial conformation of these proteins, resulting in pulling the troponin away from the binding site on the surface of actin. So troponin tropomyosin does not block the interaction between myosin and actin anymore, and the contraction results. Sorry, it's wrong spelling, contraction. We'll look on these steps of contractions more closely.